Hello, thank you for joining me. In this short video, I'm going to demonstrate how to use the ImagePro Confluence protocol. Protocols are found on the Count Size tab, and if we click on the Protocols button, the Protocols browser opens, and here we can see the protocols arranged into collections. There's the Essentials, Cell Biology, and Cell Biology Advanced. The protocol I'm looking for, Confluence, is found in the Cell Biology collection. If I scroll down here we see the Confluence protocol, and if I click Load, the protocol loads on the left of the application. I'm now going to open my dataset. At this point, I'd like to reference the source of the data I'm going to use, which is from the Broad Bioimage Benchmark collection, and this one has accession number BBBC019. And the dataset I'm using is the melanoma dataset. I've imported the images into ImagePro as a single dataset. I've used the import function, which you'll find under the File menu. There's a video for import if you're interested in importing data. Now this data is organized as a series of experiments. I've made each experiment a site in my dataset, and for each experiment there are 2 scans, each one with images acquired 20 hours apart. Now this is really Scratch datasets, but it's going to work very nicely to show Confluence. A Scratch assay is really a Confluence over time. So here we can see one image and the same field 20 hours later. And if you look at some of these different sites, you see the same pairs of image. First image and the second from 20 hours later. And I'm going to analyze the degree of confluence in each of these images. Alright, let's configure our protocol. The first thing I'm going to do is make sure that the interactive checkbox is configured. This means I can go through each step in an interactive fashion and configure my protocol. I'm also going to click on the sync button so that the protocol is aware of the channel names in my dataset. And let's click Next Step. In this step, Step 2, we get to configure the parameters used to segment our cells. So we're trying to separate our cells from our region of background. And you can see that we have 4 different parameters that we can change: threshold, smoothing, cluster size, and cluster filter. For each of these I can either edit the checkbox, use the up and down arrows, or usefully, if I hover over a field, I get a little slider that allows me to change the value of the parameter. So I'm going to set the threshold to 104. Let's change the smoothing a little bit. And I think the cluster size I'm going to reduce. And I'm going to edit the close filter as well. I'm happy with these parameters. Areas where there are cells are shown in red, and cell-free areas are unlabeled. We can see even the individual cells in the Scratch area have been found. I can now click OK. It's important to note that whenever you edit any of these settings, you should have the Show Preview Mask option selected in order to see interactively exactly what the effect of your changes will be. We can add an option to set a threshold for the minimum valid area. I think that'll be useful because I've got some very small areas of debris that I don't particularly wish to measure. So if I click on the Edit button, I can change the threshold. This is the minimum valid threshold. So everything smaller than that threshold is invalidated. And let's just set this to a sensible value. Click Apply, and there we go. Now we've excluded the noise and the debris, but we're still measuring our cells. And we can also set a reference side if we want to try and estimate the number of cells that we're actually counting. So if I click on the Select button, and then click on this area of cells, so that's a reference object with a known number of cells, in this case 2, and click OK. I can then enter my cell count there, and now ImagePro has what it needs in order to be able to estimate the number of cells in each field. So I'm going to click Apply, and move on to the next step, which is the results step. Now if we check our data collector table, we have a single table, the image table, and it contains a value of 52% for the single field of view that we've analysed. And we can see an estimated cell count of 320. And there's a snapshot that shows our area of Confluence. Now we can configure the option to auto-create a report, to auto-export results to Excel, to auto-save snapshots to the data collector, and to auto-show data collector views. This is really useful. So I'm going to automatically show a heat map, and in the heat map I want to show the Confluence percentage for each field of view analysed. I'm also going to show the block view and the graph view. And in the graph view, I'm going to set the x-axis to the collection number, the y-axis to the percentage confluence, and I've got the show all option selected, meaning that all our collections will be shown in the same graph. I'm going to hit the read button and save as, and all of our changes will be saved with the protocol. So now I'm going to click Finish and deselect the interactive button. And now if I hit Run Selected, the protocol will be run on all of the images in my set. And here you can see a progress bar that shows me the quick progress we're making. This is a multi-threaded analysis. And we can see here our heat map. Our areas with the least confluence are displayed in blue, and the most confluent areas are displayed in red. You notice that as I click on an area in the heat map, we see the corresponding image in the data collector block view, and we see the appropriate row in the data collector table. We get the same feedback from the data collector graph as well here. So if I want to see my most confluent area, I can just click on the most confluent point on the graph, and I could compare it to my least confluent areas. The final thing I'm going to do with my results is generate a report. So I just need to click on the report button, and a preview of the report is displayed in Image Pro. The report contains the heat map, the chart, the image table, which includes confluence, and a table of snapshots. This is just a preview of the table. If I click Save As, I can save it as a PDF report. And the full PDF report is generated. That brings me to the end of my Confluence demonstration. I hope you found it useful. If you have any questions, please contact Media Cybernetics and we'll be happy to help.